It's a pleasure to be here, and thank you all for coming tonight. I recognize actually most of you. I've been here a long time, more than 20 years, actually 25 years. So I feel very settled here. Seattle's great. The biomedical community here is first rate, and I really lucked out coming here 25 years ago. So I'm going to talk about expanding newborn screening. It's kind of a fusion of organic and analytical chemistry, and I guess it's the right time to talk about this project, as you'll see during the talk. There's been some recent events in the last year or two that has really added a spark to this story. I should tell you, my background is in, I've always liked chemistry and I've always liked medicine. I was going to go to medical school in college, and then I fell in love with chemistry. I had a really good high school chemistry teacher and a really good physiology teacher, so I've always been sort of half chemistry and half medicine, biology kind of things. And I've always wanted to work on something really important, and the older you get, the more important that becomes. So the time is right for this story, and it's sort of derived from what I've been trained to do and love to do. So this project is actually a team of 3 senior investigators. So myself, listed at the top, and then Frank Turacek, who's here in the audience. Frank's a mass spectrometrist professor in the chemistry department. I've known Frank for almost 20 years. And then Ron Scott is in the Department of Pediatrics, University of Washington and Children's Hospital, and he's responsible for a lot of the patients and research and treatment of rare diseases in children that you'll hear about tonight. And then the UW list are graduate students and postdocs that have worked, the key people that have worked on this project in my lab over the last 7 or 8 years. And we've been working in the last 4 or 5 years with Enzyme. It's a company in Boston, and Joan Koitzer is the key person involved, and they've been key in translating this work from the bench to the real world of medicine. And the funding is from the NIH, from Enzyme, and from BioMarin, which is another biotech company. And I'd like to dedicate the seminar to Nestor Chamolas, who he passed away a few years ago. He was running the newborn screening facility in Buenos Aires and was the first to show that you could measure enzyme activities relevant to newborn screening in dried blood spots. And that'll become important as we go through this talk. So a very seminal discovery. So let me tell you the truth about this story. Really, it's the truth. And in 1985, my wife and I, we had our second child, and my wife was over 40, so she had amniocentesis, which is usually done if you're older. And the nurse came in and did the procedure, and I asked the nurse, "What do you do?" And she said, "Well, we look for spina bifida. They look for a protein in blood, and they look for Down syndrome. They look at the chromosomes." And I sort of said, "Well, that's all you do?" And I began, I don't know, I think I saw that movie Lorenzo's Oil about a family that had a baby with a rare disease, and the father was committed to trying to find a cure. And I'm trained as an enzymologist, and I thought about, right at that moment during the amniocentesis, I thought about why not test for all these enzymes that could be deficient in these babies. And I was very naive about why not test for all of them, right? And I imagined a technique of using mass spectrometry of being able to assay maybe 200 or 300 different enzymes. So why not just, instead of just spina bifida and Down syndrome, why not assay for all 200 of these things? And then quickly you learn that that's not really a good idea, and you'll see why in a minute. But I thought about a technology that would be able to assay as many things at the same time as possible, and it should be very easy. So I had this idea of using tandem mass spectrometry, which you'll hear about for newborn screening. So I contacted the local expert on genetic diseases in babies, and that was Ron Scott. He's at UW Medical School and Children's Hospital. I had lunch with him in 1995. So I put this on the back burner for 10 years. Ideas pop into my head all the time, and there's just not enough time to work on all these things. And so for 10 years I thought about it, and then I was ready to talk to Ron about it. And I think I was in a good mood one day, and I said, I need to do something. I need to expand my program. And when you're in a bad mood, you tend to want to reduce the size of your program. And I was in a good mood that day, so I thought I'd have lunch with Ron. And he got all excited about this mass spec stuff. So then I figured we should recruit Frank Turacek, who's the local mass spec guru in Seattle, and the three of us became a team. So then we needed a graduate student, and that was Scott Gerber in 1998. He's an analytical chemistry student, joined the lab, and he started working on this for the first time, brand new project. And he kind of validated the use of mass spectrometry to study an initial set of enzymes that I won't talk about tonight. That was early days. I think a key thing happened in 2004. Graduate student Eugene Lee in chemistry applied tandem mass spectrometry to analyze 5 different lysosomal enzymes using dried blood spots, and you'll see the significance of that in a minute. Another key happening was a year later when Joan Koitzer at Genzyme made a commitment to develop these University of Washington assays for worldwide newborn screening. I mean, you can always publish something in a journal and it just sits there. You really need industrial commitment to to bring the ideas to fruition. Otherwise, it just sits in the library and collects dust. And it's not trivial to bring things from the bench, to the real world. It takes a lot of energy and commitment, and I certainly don't have the time to do that. So I'm very grateful to Joan and Genzyme for their efforts. So they made a commitment at Genzyme to synthesize the reagents that you'll hear about. They were approved by the FDA. And then newborn screening, it comes under the umbrella of the CDC in Atlanta, that you all know about, and they distribute the reagents to newborn screening labs worldwide for free. I'll tell you, in 2008, the Wadsworth Center in New York initiated newborn screening for Fabry Disease, and you'll hear more about that. And then later, Taiwan got into Pompe Disease. Illinois is moving forward with 5 more diseases. And in Washington State, we're going forward with 3 of these diseases. You're getting the feeling now that newborn screening, at least in the United States, is state by state. Every state a little bit different. It's under state law, not federal law. And there's some talk about trying to make everything the same, but right now it's state by state. So that's why you see some of these stories starting in certain states and then moving to other states. So the point of this is it started from this amniocentesis with me and my wife when my son was almost born, and sort of mulled around in my head, and then I found the right people at UW, got the students involved, and went all the way through the process with industrial support all the way to the real world of medicine. So what are lysosomal storage diseases? It's a group of about 39 distinct genetic diseases, and they're mostly due to a deficiency of an enzyme in the part of the cell called the lysosome. These enzymes are involved in degradation of molecules that the cell no longer needs. So if you have a broken enzyme, the cellular component accumulates, and the lysosome, the trash can fills up and it's never emptied. And the lysosome swells and the cell dies, and that's a problem. As a group, it's about 1 in 5,000 births, so it's a little bit more prevalent than PKU. You've all heard of PKU. That's the disease, when you can't eat NutraSweet. That's the inability to break down phenylalanine. And it's an interesting story. So PKU is sort of the hallmark of newborn screening. You see, the idea is these babies are born and there's something wrong with them, but they don't know exactly what, because this disease is very rare. And by the time they figure it out 2 or 3 years later, the baby is in really bad shape. And if only you had known the baby had PKU, you could treat the baby simply by eliminating phenylalanine from the diet. But by the time you figure it out, it's too late. So a guy named Peter Guthrie had the idea of maybe screening for babies randomly and find out at birth whether they have this disease, and then talk to the parents. So that started the newborn screening game maybe 30, 35 years ago, 40 years ago. So the key is you need to have a treatment. We're not going to screen for everything, although there's some value in that. But the idea is that these lysosomal storage diseases, many of them have become treatable in recent years. And it's the case that these are rare and they're misdiagnosed, so the parents go through agonizing pain trying to figure out what's wrong with their kid, going from doctor to doctor. By the time they figure it out, it's too late to start treatment. Treatment doesn't work very well when the baby is almost finished. And fighting this disease. And so it makes sense when there's a treatment to find it early for lots of reasons. I guess one way to put this, I go to these meetings where you have parents with these sick children attend and scientists, and we always have discussions about the value of newborn screening, and we always take a vote, and the vote is always essentially unanimous, like 600 to 0, that we should have newborn screening for these treatable diseases. So at least the parents of these sick children, they all want newborn screening. Whether or not the general public wants it is a little bit more controversial. But anyways, it's always by consent. It's usually by consent anyway, so you can choose whether you want it or not. So the key is that early detection in newborns is warranted for treatable lysosomal storage diseases in cases where early treatment leads to better outcome of the clinical outcome. Now, there was a movie that just came out a month ago called Extraordinary Measures. Probably none of you saw it. It didn't do very well. It stars Harrison Ford and Brendan Fraser. Those guys are pretty famous. It was more of a Hallmark sort of Sunday night movie rather than a big screen movie. So my lab, we planned to go see it at the Neptune, and by the time we got there, they had canceled the movie. So then we went to the Varsity the week later. Then they canceled the movie. So finally, the only place left in the Pacific Northwest was the Supermall in Auburn. So we went to the Muckleshoot Casino for a drink, and then we went to the movies. And this is 9 o'clock on Saturday. I have a picture, it didn't come out, but we were the only people in the movie theater except the guy showing the movie. And if we took him out, we probably could have watched the movie twice if we wanted. But anyway, so we were the only ones there, right? And next to it was Avatar. So it's a very good movie, actually. And the guy who made the movies about, John Crowley, is actually coming to UW for a seminar next year. And I've got an invitation out to Harrison Ford and Brendan Fraser, so we'll see if they come. But that'll probably get a good turnout. So it's a true story about John Crowley, who he was a business executive at Bristol Myers Squibb, and he absolutely true story. He had a couple of kids, and when they were about 2 years old, they were diagnosed with a devastating lysosomal storage disease called Pompe disease. And he was a little bit frustrated with the way research was going, so he decided, he found a guy in Oklahoma that was doing research in mice on Pompe disease for the treatment. And so Crowley invested his own money and left his cushy job at Bristol Myers Squibb and started this company with this scientist in Oklahoma, and invested his own money, raised a lot of money to work and try to find the cure for Pompe disease, in part to use on his own kids before they die, because this disease in infantile form is always fatal. So the company did very well, and then Enzyme finally bought the company and took the drug through clinical trials. And about 3 years ago, the drug was approved. It's called Myozyme. Then Crowley had to quit the company so that he could use the drug on his own kids. Otherwise, it's a conflict of interest. So he did that, and his kids are doing much better, but not great because his kids were diagnosed late. And the kids that I'll show you at the end of the talk that are diagnosed early using our technology do really well if they're caught early. So the punchline, I guess, is you need the drug. That's half the story. The other half of the story is these diseases are so rare that the treatment is really not that useful unless you have a way to find the babies. And the only way to find them really is newborn screening. So that's what I'm going to tell you about. So the lysosome, the treatment is called enzyme replacement therapy. And what they do is, it's one of the triumphs of the biotechnology industry, right? The molecular biology allows one to make large amounts of proteins in bacteria, for example. And one of the uses of these proteins is you can make the enzyme that's deficient in these babies, and you can infuse the enzyme into the blood of these babies every 2 or 3 weeks. And the reason it works for lysosomal enzymes is because cells naturally swallow the fluid outside of the cell, called endocytosis. And the components, which include the recombinant lysosomal enzyme that you give, is then transported into a vesicle, which then ends up in the lysosome. So the problem with getting a protein like an enzyme inside of the cell is it can't really cross the cell membranes. But through the process of endocytosis, which is a natural process in the cell, the protein is taken up and delivered properly to the lysosome. So this works for lysosomal enzymes, but it doesn't work for enzymes in general. So the point is that you can inject an enzyme into the blood, and it's taken up by cells and delivered to the proper part of the cell where it can function. So you can just replace the enzyme by giving a correct copy of the broken enzyme that's broken in the baby. Now, in Pompe disease, it's a disease of the muscle because the enzyme that's missing is involved in the breakdown of glycogen, which is a form of energy storage in muscle. It's a type of muscular dystrophy. When these babies are born, their hearts are filling their entire cavity. The heart doesn't pump very well, so it gets really big. And after you treat with the enzyme replacement therapy, you can see the heart returns to its normal size. The muscle, the microscopy of the muscle returns to a normal morphology. The EKG returns to normal. And I'll show you a movie at the end where the baby born can barely breathe because the muscles aren't working, can't walk, can't move his head, and then all of a sudden the baby is much better. So here's the problem with these diseases. When you have the infantile form of Pompe disease, the first symptoms occur maybe 1 year after birth, and then diagnosis is made at age 5, and the babies are dead a year later. So by the time the point of this slide is by the time the disease is diagnosed, the babies are almost gone. So we have this treatment, but it's of no use unless you can catch the babies early. That's the whole point. So let's talk about our story at University of Washington. Now we're back to tandem mass spectrometry. So what is tandem mass spectrometry? I'll show you in a minute. But the key advantages are that tandem mass spec can be used to detect specific metabolites, enzyme products in this case, in complex biological samples, for example, blood, without having to do time-consuming separation techniques. It's a technique, it's the perfect technique for finding the needle in the haystack. That's literally what it does. And because every enzyme product can be distinguished in the mass spectrometer, we can detect many such products simultaneously, and this allows us to assay many enzyme activities at the same time. And it's a very sensitive technique. We only need a couple of microliters of blood or maybe 1,000 cells. And you'll see that in a minute. It's very sensitive. It's highly quantitative. If we're going to measure the enzyme activity, we have to do it. It's a numbers game. We want to know, does the baby have 2% of the normal activity or 100% or whatever? It's a numbers game. And we can make the reagents. They're easy to make and they're inexpensive. It's a very fast analysis and very easy, which is obviously important for screening hundreds of babies that are born in Washington State every day. So the idea is an enzyme, operates on a molecule called a substrate, S1 in this case, operated by enzyme 1 to convert it, a chemical reaction to make the product P1. And if you have, in this case, 4 enzymes, you could have 4 reactions occurring at the same time. So if we add the mixture S1, S2, S3, S4, a mixture of 4 substrates as a cassette, as a single mixture, the enzymes can convert all of these simultaneously into the mixture of products P1 through P4. And if we can detect all of these simultaneously by mass spec, we've assayed all of these enzymes at the same time. So the idea is to let the sample incubate for a certain amount of time, to let all of these substrates convert to the product, measure the amount of product, and then the amount of product divided by the amount of time is the velocity of the reaction. And the velocity of the reaction is proportional to the activity of the enzyme. Now, we have to make this quantitative, and to do that, it's very, very easy. What we do is we add, in the case of P1, we add an analog of P1 that's chemically the same, but it has a heavy isotope in it, so that P1 heavy and P1 not heavy can be separately quantified in a mass spectrometer. And the key is we know exactly how much P1 heavy we add because we added it to the sample. We add a known amount. So if the mass spec signal for P1 light and P1 heavy are the same, and we know we added 100 picomoles of P1 heavy, we know that the amount of P1 light is also 100 picomoles. So that's called an internal standard. And think about the beauty of that. So if some of the P1 is lost because it sticks to the walls of the test tube, well, the same thing happens to P1 heavy because they're chemically identical. So whatever, it doesn't matter if some other enzyme consumes some of the P1, it does the same thing to P1 heavy. If the mass spectrometer hiccups during the detection of P1, it does the same thing for P1 heavy. It doesn't matter, as long as we take the signal for P1 and P1 heavy, compare the two, and we assign that signal to P1 heavy, we know the amount of, we assign the signal for P1 heavy to the amount we added, we know how much P1 is there. So it's a very simple solution to make the method extremely perfectly quantitative. Now, mass spectrometry of big molecules like these enzyme products is a bit of a problem in the old days. Because what you have to do in the mass spec is you have to get the molecules to go into the gas phase. So for water molecules, that's easy. You put them in a pan and you boil the molecules, and you give the molecules enough kinetic energy that they fly into the gas phase. So bigger molecules don't do that because the temperatures you need to get them to fly causes them to fall apart. You need to heat it too much. So what we do is we trick the molecules. The molecules aren't very smart. We're smarter. We can trick them. We can make them do things without them thinking about it, right? That's called tricking them. And so what we do is that's exactly what we do is we trick them. So we take a stream of liquid water, which have these molecules in them, dissolved in them, and we send it through a small tip of a little port in a needle, and little droplet of liquid with these molecules emerge, and we blow across that droplet a high stream of nitrogen gas. And we break the droplet into little teeny droplets. It's called a nebulizer. It's like the thing you put in your hotel room when you sleep in the Rocky Mountains and your nose is all, dried out because the humidity is humidifier, right? So we break the little droplets into even smaller droplets, and then we put this under high vacuum so the water gets evaporated, and the molecules are left in the gas phase. They have no choice, right? We've sent these little droplets into the gas phase, and we take the water away. So they're flying, and they're saying, wow, that's not so bad. I can do this, right? And they have no choice. They're flying around as naked, separate molecules in the gas phase. They're flying. They're in the gas phase. No problem. And then we can send them through a mass spectrometer and measure how heavy they are. We can measure their mass. Okay? So we can trick them to be in the gas phase. Now, all right, so then we put them through a mass filter. So we say if you have a certain mass, a certain weight, you get to fly through the center of the tube and reach the detector. But if you have a different mass, you don't go through the center of the tube. You crash into the wall and burn. Okay, so only molecules of certain mass make it through the tube. So that's fine. So if our P1 that we want to detect has a certain mass, we just filter out so only P1 goes through and nothing else goes through. The problem is if we do this in blood, there's probably different kinds of molecules in blood, different molecules that happen to have the same mass. All right, that's bad. So what we do is we send them through a chamber where they collide with argon molecules, and we break these molecules into smaller molecules. And then we send those small molecules through another mass filter, and we insist that only molecules of a certain mass go through that second filter. So now if you have two molecules that happen to have the same mass that go through the first filter, when they collide, the chances are they're going to have different masses. Their fragments are going to be different, and only one of them is going to get through the second filter. So we're insisting, we're only looking for molecules that have a certain mass and then a certain fragment mass. It's a double selection, right? And by the time you do a double selection, the molecules that make it through are only the ones you want to analyze, P1 or P2, but nothing else in blood. So it's really a way to find the needle in the haystack, and it's very easy and it's very fast, and it's very sensitive. The other thing we need to be able to deal with is what we have available in newborn screening labs is not, fresh blood. I mean, it can't be heroic, right? We're going to take 200 babies a day in Washington State, and every baby that's born, we're going to analyze their enzymes. We're not going to take fresh blood and put it on dry ice and send it in a styrofoam box with FedEx, hope it gets there in time, to the newborn screening lab. No, we can't do that. It's too heroic. What we're going to do is we're going to put a little bit of blood on a piece of paper, let it air dry, and put it in the mail and send it. And much easier, right? It's sort of like voting. If it's too hard to vote, nobody votes, right? It's the same thing with newborn screening, right? if you just say, well, let the next guy vote. My kid's not going to have this rare disease, so I'll just skip it, right? So it has to be really easy if you're going to screen a lot of people, right? So it turns out these lysosomal enzymes, they're perfectly happy dried down on this piece of paper. We take a little 2 millimeter punch of that dried blood spot, so little teeny, about a microliter of blood, 1,000th of a milliliter. And we put it into a tube and we give it some buffer, and the enzymes are fine. They just leach off the paper and they dissolve in the buffer and they're fully hydrated and they're fine. And the buffer has a bunch of substrates in it, all that S1, S2, S3, S4. We let it incubate. We simply shoot it into the mass spectrometer and measure all of the products and all of the internal standards more or less simultaneously, actually in a matter of 20 seconds per sample. And we know exactly how much product is formed, and we know how long it took to form the product, so we know the activity of all the enzymes simultaneously. So let me, so here's a little chemistry. This is one of the enzymes, Niemann-Pick disease. You're deficient in an enzyme that takes this lipid called sphingomyelin and cuts the phosphate backbone, makes a lipid called ceramide. So we measure the amount of that ceramide there. That's formed enzymatically from the top molecule, okay? And if the amount of ceramide you formed over a period of time is the activity of the enzyme. And if you're deficient in this enzyme, if you have a low activity, you have a disease called Niemann-Pick disease. You're missing this lysosomal enzyme. So the trick is in your blood, you'll have these ceramide lipids already naturally. So what we do is we use a ceramide that's a little bit different than the natural product. It has a truncated carbon chain. It's not natural, it's man-made, person-made. And it has to come from the top molecule because there's no ceramide in blood that has such a short carbon chain, okay? But the enzyme doesn't care so much. And the internal standard is almost the same thing, but it has 2 less carbons in the carbon chain, so it has a different mass, right? It's different mass, different size molecule. But they're almost the same, so they behave the same in the mass spec, and everything's the same. We know how much of that bottom molecule we have, so we get the signal from the top molecule, get the signal from the bottom molecule, from the ratio of those 2 signals, and multiply the amount of bottom molecule we have. We know how much top molecule we have. It's very easy. So here's another, so the signal is very easy. On the left is just blank filter paper without blood. You see the internal standard because we added it intentionally, the peak to the right on the left graph. And there's no peak for the product. There's a little blip because there's no blood. And then you have a healthy person. You see in the middle graph, the internal standard to the right, the equal size, almost equal size peak, enzyme generated product. And then you have person missing the enzyme. You see the internal standard on the far right graph. You're essentially missing the product generated by the enzyme. So in one case, no blood. In the other case, blood from a healthy person. On the right, you have blood from a person that doesn't make that product because the enzyme is deficient. Here's another disease, Fabry disease. It also hydrolyzes a lipid. It releases a ceramide. At this time, it releases a galactose moiety. We can differentiate that ceramide in the middle from the previous one because we just use a different length carbon chain. It's very easy. So we can mark this process. Here's another disease called Fabry disease. This guy has a few tricks in it. When we bombard these molecules with argon and break them into fragments, we don't want them to break into lots of tiny pieces. We want them to break in one place. Why is that? Well, if they break in one place rather than a bunch of places, the assay is more sensitive. So if you take a molecule in the gas phase and you break it into 100 different pieces, you're going to be 100 times less sensitive than if you break it into 2 pieces, right, or 50 times less sensitive. So how do we make it break in one place? We put a chemical entity in the molecule that's very easy to break. It has a weak chemical bond. So we direct the fragmentation to occur in one place. And that just makes the assay more sensitive, okay? And in this case, on the bottom, the internal standard, we use deuterium. You can see that. It's chemically identical to the product that we analyzed, but it's heavier in the mass spec because it has the heavy isotope of hydrogen called deuterium. Pompe disease. It's the same as the Fabry disease. It's a different sugar. We differentiate this, but you see the linker, the carbon chain is a little bit longer than in Fabry. Pompe disease, the topic of this movie. It's a little bit more complicated than measuring this activity. What plagued the field for more than 30 years is that there's 2 enzymes in blood that catalyze the same reaction. One is the lysosomal enzyme that you want that's relevant to Pompe disease, that's deficient in Pompe disease. The other enzyme is on the surface of blood cells called neutrophils that has the same activity and is not deficient in Pompe disease. So you have two different enzymes catalyzing the same reaction. Only one of them is deficient in Pompe disease. So if you don't deal with this problem, you could be deficient in one enzyme and not know it because you have the other enzyme. So we had to find an inhibitor that blocked the neutrophil enzyme, so it just wipes it out. And then the only activity that you're left measuring is the Pompe enzyme, the lysosomal enzyme. And that was the breakthrough that allowed Pompe disease newborn screening, the discovery of this inhibitor called acarbose that blocks the neutrophil enzyme. So sometimes, another problem we have sometimes is the synthesis of the enzyme substrate is problematic. So organic chemists are very good at making complicated molecules. They're just not very good at making complicated molecules well. It's no picnic for the bacteria to make these molecules either. They have to do it in 35 steps. So either we do it in 35 steps or the microorganism that makes the molecule do it in 35 steps. It's no picnic for anybody. So what we have to do is the synthesis of some of the substrates can be problematic. And that was the case for a disease called MPS II. It's an enzyme that removes the sulfate from one of the OH groups of the sugar. And it's a bit of a mess to put the sulfate on only one of the 4 possible hydroxyl groups. So we had to come up with a synthesis to do this in only 4 steps because every year Genzyme has to make about 10 grams of this to support worldwide newborn screening. Now 10 grams is a trivial amount compared to some pharmaceutical that millions of people take that you have to make in hundreds of kilograms. But 10 grams is no fun to make if it's 35 steps, and it's kind of a deal breaker. So even a compound that you need in only 10 grams per year, the synthesis has to be practical. So we spent a lot of time optimizing organic synthesis, and that's a very big part of this project. And that's why at the beginning of the talk I said fusion of analytical chemistry and organic chemistry. I don't want to trivialize the organic chemistry. A lot of the time we spend on this project is organic chemistry. So this is some of the data that we generate. We generate very small amounts of data because we don't have access to lots of dried blood spots. But you can see on the right you have, this is the Krabbe assay. Infants, we had 15 infants, 16 adults. They have a range of activity that's well above the activity of the 12 patients that have Krabbe disease, you see on the far left. So in terms of our assay, it works pretty well. We can easily distinguish patients from normals. And the point I want to make is that normals have a range of activity. We're all different from each other. We all have different amounts of activity. We have polymorphisms in our genome. And we all, it's not a black and white thing. It's not like you have 100% of the activity or 0%. Normal people have a range of activities. Affected people have lower amounts of activities. And the less you have, the more you're affected. Another way to say that is the less activity you have, the earlier onset is the disease. And that's an issue because if you get this disease in your 60s, it's not as big a problem as if you get it when you're 2 months old, right? So the amount of activity it's important, and it's not binary. It's not, yes, you have the disease, no, you don't. It's a gray scale. And that's one of the problems in newborn screening is the scale is gray. When is it a disease and when is it not so bad? And so now let's talk about translation to newborn screening. So Genzyme, as I mentioned, through the efforts of Joan Koitzer and others, have taken our reagents that I've talked about tonight and have made these reagents in a facility in Switzerland under GMP conditions. GMP is good manufacturing process. This is a process that's you adhere to certain rules for making things so that you can ensure good purity. This is something that the FDA insists on. You have to keep notes, you have to keep a lab notebook, and you have to wear lab coats and keep your fingers out of the flask, and all the things that graduate students don't do, right? When you make the stuff for real, you have to be careful, right? And it has to be done properly, right? It's like cooking your own food at home versus a restaurant. There are rules, right? And then the reagents are registered with the FDA as something called Analyte-specific reagents. And then the newborn screening lab can take this Analyte-specific reagent that's approved by the FDA and put together sort of a home brew. They can get our assay working in their lab using the appropriate robots or whatever they want to use. They have freedom to do whatever they want. The only thing is that the reagent, which is the key component, has to be FDA approved. The CDC is responsible for newborn screening in the United States. They provide all the training for the newborn screening centers and distribute the reagents to the centers. And I should point out that Enzyme distributes these reagents that were developed in my lab to newborn screening labs free of charge, right? The other side of the story, of course, is they sell then the drugs that are used to treat these babies, but of course that's not a bad thing, that's a good thing. Now, each newborn screening lab will adopt the Tandem mass spec assays using the equipment available in their lab, as I said. And then medical doctors will intervene when a baby in jeopardy is found. Okay, so now it sounds simple, it's not so simple. First, we get into politics. Now, politics of newborn screening is definitely there, right? It's a state-by-state game, right? So whoever, sometimes it's grassroots, often it's parents of these sick children form a foundation, lobby, governor of that state then approves. And this certainly happened in New York. Jim Kelly, a famous professional football player, Hall of Famer, his family had a baby with a devastating disease, Fabry Disease. And when the treatment, they started a foundation called the Hunter's Hope Foundation. That's a good thing. And it's basically a support group of parents that have these sick children. It's kind of something I would want to do if I had one. And they get together and tell stories, you know, just like other kinds of problems. And they lobbied hard when the treatment for Fabry disease became available. They lobbied hard for newborn screening. And Pataki, the governor of New York at the time, decided to make it law to expand newborn screening in New York to include Fabry disease. Now, this is a little bit controversial because Fabry disease is very rare, and it was not necessarily next in line of priorities for expansion of newborn screening. So this one was pushed to the front a lot, in part because of grassroots efforts from this family. Now, okay, that's okay. I don't know, it has its good points and bad points. Fabry disease screening is not moving quickly worldwide because people think it's too rare. So this is happening in New York right now. A lot of places are watching. We'll see what happens. I think, for example, in Washington State, Ron Scott would say that I work with, that Fabry disease is not going to make the short list of things to screen for, right? There are other priorities. So it's political, it's controversial. The New York Times finds it controversial. I should say that my group, or myself, I have no say in what we screen for. Our job is to develop the best assays we can, and we leave the decisions to parents and to doctors and politicians. I mean, I couldn't possibly have anything to say about newborn screening for a disease. I don't have a sick children. I'm not a medical doctor. I don't have the resources to make these decisions. I have no say at all in what we should screen for, and none of the people in my lab do. Our job is simply to, I really believe that. I mean, people say, people say, well, you've created the tools that now create the problem, and I say, well, my job is to create the best tools we can, right, and not to impart any decision on what to screen for. So, I feel that way. All right, so now you don't just go live with a newborn screening. You don't just do it on babies as they're born, because this disease is sort of like 1 in 100,000 babies, and if your screen shows potential disease in 1 in 100 babies, you have a big problem. You have a big false positive problem. Think about all the anxiety among all the mothers and fathers. Think about all the callbacks. Think about all the return visits of the babies and the parents and all the follow-up studies. 1 in 100. Every week you'd have 7 babies and only 1 in 100,000 have the disease. So you don't go live until you know you don't have this problem. So what New York did is they took 150,000 blood spots off the shelf from babies that were already born, and they got a remarkably low, the bottom line is they got a remarkably low false positive rate. So based on this, I won't go through the numbers, they decided it was time to go live. They were very impressed with this assay. So they went live, and they've been doing this for about 2 years, and they've found only about 4 or 5 babies. So it turns out to be a little bit rarer than we thought. These babies are doing pretty well, so we saved the lives of 3 or 4 babies, that's a good thing. But there are other diseases that are more prevalent that probably have higher priority. But to the Hunter Hope Foundation, this was a big priority, and I understand their point of view. So then a guy, Bob Evanowski in Illinois, had some sick children. This was a really sad story. His kids were misdiagnosed, and they thought they had cerebral palsy. They turned out to have something called metachromatic leukodystrophy. By the time they figured it out, they had 2 more children, identical twins. This is a male disease. So the twins so now he's got 3 kids with metachromatic leukodystrophy. If he had known what the first kid had, he could have maybe had some Planned Parenthood for the twins. The other thing is that the first baby was born before the treatment. The second babies were born after the treatment. They were treated early. They're doing fine. The first kid is not doing so good. So obviously he became a big fan of newborn screening, and he lobbied the governor of Illinois, who was this guy named Rod Blagojevich, who is now on reality TV shows. But so one of the good things he did was expand newborn screening in Illinois. So they're now mandated to do 5 of these diseases. This is the first multiplex lysosomal storage disease newborn screening anywhere in the world that will start in about 2 years. For better or worse, they're going to do this. So Taiwan, I think, is the most successful story I can tell you about tonight. So Pompe Disease, this movie that I talked about, this devastating muscle disease, is more prevalent in Taiwan. So it was a priority in Taiwan to screen for Pompe disease. And using our technology, they started the screen about a year ago. Several other lysosomal storage diseases are being piloted in Austria, Italy, and here in Washington State with people in my lab. And the newborn screening lab is just up the street in Shoreline. Mike Glass is the director. We're piloting now a triplex assay for 3 of the lysosomal storage diseases that Ron Scott thinks are the highest priority. The ones that show the best response to treatment, the ones that are not the rarest, the ones that make the most sense in his opinion. So the other newborn screening labs are watching closely. So let me end with a movie. This is a movie from Peter Su, the guy in Taiwan who, so in the past year they found about 24 babies with Pompe disease, infantile form. This is a very terrible disease. The babies are born, they have almost no motor control. You'll see this baby won't be able to follow any moving object. Eyes are closed most of the time because you can't open your eyelids. Baby will be put on a ventilator to breathe, eventually on a respirator, and then eventually on a ventilator, and eventually die within 6 months. So you're going to see this baby at birth who won't be able to move his head and follow the toy. And then you'll see the same baby about a year and then 2 years after the establishment of enzyme replacement therapy, treatment with Myozyme. And I should point out that this baby wouldn't have been found in time without newborn screening that we developed. So we take some credit for these developments as well. So you need the treatment and you need the screening. You can't have one without the other. It just doesn't work. So yeah, I think the movie needs no narration. So none of the things this baby is doing he could do without this treatment. So. Yeah. So I don't know much about this disease really. I'm showing you this movie from Peter Su, but I think it's pretty impressive. Okay. So the Extraordinary Measures, the sequel. I'd like to thank some key people in my lab and around the world. So Scott Gerber, I mentioned, was the graduate student that was the first one to work on this project when Frank Turchak and myself and Ron Scott got together as a team to start this. And this is, so Scott's a professor at Dartmouth Medical College in New Hampshire. Tricia Duffy is a postdoc in my lab who has developed the triplex assay that's being piloted in Washington State as we speak, and the data look really good. And we will go to Ron Scott will go to the legislature soon and propose the first triplex assay for these 3 diseases anywhere in the world, and everybody will watch and we'll see what happens, and maybe it'll spread to the whole world. Brian Wolf is a graduate student in my lab, working also on these assays. And I wanted to mention Bob Voigt at the CDC, who we've met a few times, who's responsible for newborn screening, and Victor De Jesus, also at the CDC. These are the two key guys. CDC is a very big place. These are the two key guys that have launched this project at the CDC. And this is Joan Koiser, I've mentioned several times, at Enzyme, who, the visionary who has, it's kind of an interesting story because Enzyme developed the treatment, and they saw it as a conflict of interest to go out and develop the technology for finding the babies that need their drugs, right? That's kind of a conflict of interest. But then they had these drugs, and they had no patients because they had no technology for finding the babies, so it seemed kind of silly not to do something about it. So they had started to work on newborn screening as well, but they latched onto our technology because I think it's really the best in the world, and they have then taken our technology through the process of manufacturing reagents and making it happen. I mean, we don't have a company here at University of Washington. We're too busy for that. My chairman is always bugging me about that. We give these reagents away for free. My chairman is always bugging me about that, but I don't feel so bad about it. I don't know. The university pays me pretty well. It's okay. And really, this has been very gratifying. I mean, at the end of the day, you go home on your bike and you're smiling and it's dark and it's raining, and no awards committee or politician or grant panel can take that away from you, right? And this is why it's really fun to be a scientist. At the end of the day, to be happy about doing something like this, and that's the only thing in my mind that matters, and that's why this job is a really great job. So I think that's it, and I'll be happy to answer any questions, and thank you all for coming. So
I was wondering if you could repeat the cell processes you described at the beginning that cause this cellular malfunction.
Yeah, so there's this compartment in your cell called the lysosome, and it's kind of like the trash compactor or trash. It's the place in the cell where things are taken for destruction and removal from the cell, and there's a series of enzymes that are involved in chopping down these metabolites that are no longer needed. These hydrolytic enzymes, and if you're missing one of those enzymes, this metabolite accumulates, and the lysosome swells, and eventually the cell dies. So in the case of Pompe disease, we're talking about an enzyme that's involved in the degradation of glycogen, which is the major energy store in muscles. So this is a disease mostly of muscles, okay? Because you have a lot of glycogen breakdown in muscles. So the muscle cells are constantly dying because you can't empty the trash can, and the muscles stop working, and you need your muscles to pump your heart and all that stuff, pump your blood. Some of these diseases manifest in other tissues. So the Tay-Sachs disease, for example, is a disease mostly of the central nervous system because the molecules that are broken down, the enzyme that's deficient works on molecules that are prevalent in the brain. So all cells have these lysosomes, and lysosomes break down lots of molecules in all these cells, but certain metabolites are more prevalent in some types of tissues than others. And so some of these diseases manifest in certain tissues. Tay-Sachs disease in the brain, Pompe disease in muscles. So you're just basically missing an enzyme that you need to chop molecules down to get rid of them. Yeah.
Thank you.
Yeah, one slide you talk about the using internal standard using
Yes.
heavy peptide in
Yeah.
enzyme digestion.
Yeah.
I think you assume that the efficiency of enzyme digestion is constant. I think in reality, the activity of enzyme may not be constant from one batch to another batch. I don't know how you control this kind of issue.
So the only thing that matters is we've shown for these assays, I didn't show the data, but we incubate the samples, let's say overnight for 12 hours. And it's important to show that the product of the enzyme is constantly being made over that 12 hours. And we've shown that the product accumulates linearly over the 12 hours. So we know the enzyme is working at the same efficiency constantly over the 12 hours. So if it's linear over 12 hours, it's perfectly valid to take the total amount at 12 hours and divide by 12 hours to get the velocity, to get the slope. So the enzyme, we've validated already that over the period of the assay that the enzyme in any particular blood spot is operating at a constant velocity.
Okay.
And whatever happens to the internal standard happens to the product the same because they're both present together. And it doesn't really matter what happens as long as there's some left at the end for us to detect. Yeah, and that's the beauty of the internal standard is it accounts for all losses of the analyte we're trying to detect because the molecules are chemically the same, and so they suffer the same fate. And whatever happens, we don't care as long as we can see something at the end. But the key is to show that since the product accumulates linearly over the time period of the assay, that it's okay to take the endpoint and divide by the time. If the enzyme pooped out after an hour, that would not be good. You'd have to do it inside of an hour then, but that's not the case. Yeah.
Any racial or regional differences in the frequency of any of these as far as you know, or is it too soon?
I'm not an expert, but certainly there are. You know, Tay-Sachs is very common in Ashkenazi Jews, and so in New York State and in Israel, they talk a lot about screening for Tay-Sachs disease, even though there's no treatment, you know, because there's carriers, you can maybe Planned Parenthood. Pompe disease, I mentioned, is more prevalent in Asia, especially Taiwan. I'm not an expert, but certainly these are rare events in gene pools, and there certainly is segregation throughout the world. And so, yeah, that has to be taken into account, you know, where to screen, what to screen for and where, yeah. And I'm not an expert, but there certainly are regional differences, yeah.
Thank you very much.
Newborn screening is currently done in the state, and you know, there's using many of the techniques that you described, with
Yeah.
tandem mass spec, but they use the whole strategy is similar but different in the sense that they use the naturally occurring agents that are already present in,
Right.
in the sample.
Right.
Is that, you've made it, you know, what you've done is really done something quite different, and that
Yes.
is to take advantage of the enzymatic activity
Yes.
that's resident in the cells that
Yes.
are there, which implies that you can't do it the other way. Is there a reason that you can't? Are do the cells not give out adequate stuff? Can you not measure the breakdown products?
I
of
think that's a very
enzymes?
good question. So tandem mass spec has been used for the past, I don't know, 5, 6, 7 years for screening metabolic diseases, including PKU. The idea is if you can't break down phenylalanine, for example, the amount of phenylalanine amino acid in your blood will be elevated. So if you just analyze for all the amino acids, and that's the beauty of mass spec, you can do them all at the same time, you can see which metabolite accumulates because the enzyme that breaks it down is missing. So in principle, we should be able to spot these lysosomal storage diseases by looking for an accumulated metabolite. So the substrate of the enzyme should accumulate, and we should see that. And what we're doing is we're not doing that. We're taking blood and we're adding substrate and we're letting the enzyme act on the substrate to make the product. We're measuring the amount of product. So we're doing an enzyme assay rather than looking at, in a sense, the result of a deficient enzyme in terms of an accumulated substrate for the enzyme. And the reason people have looked hard for these accumulated metabolites in the blood for these lysosomal storage diseases, and there has not been reliable. It's simply, there's just nothing reliable that marks the disease. And so everybody agrees now that the best thing to do is just to measure the enzyme directly. So there are indications in Pompe disease that certain glucose tetrasaccharide accumulates, but it hasn't been very reliable in the end for reasons that are not clear. So it just hasn't panned out that you can look for accumulated metabolites, and that's not the most reliable way to spot these diseases. Enzyme assay seems to be more reliable. There's a better correlation with the disease. But it's not obvious why that is. I mean, why, yeah. Maybe the lysosomal metabolites don't leave the cells very well, whereas the amino acids are already in the blood, right? I don't know, yeah. So the key is we're looking at, we're using blood and we're not using cell lysates and things like that. So,
Do you have a sense of how many plexes you could multi in this?
Yeah, Frank could maybe, I think easily 100, maybe 200. I think it's no problem. So
Because
there's enough.
Each substrate is different, and so you
Yes.
might be able to do it in such a
Yeah,
way.
so the problem comes, can you find a buffer that keeps all of the enzymes happy? And the answer is no. You can, to some extent, you can consolidate the buffer. I don't think it'll be possible to take one reaction with one buffer with 100 substrates, shoot it. I think you could analyze 100 metabolites in that solution easily, but I don't think you'll find a single buffer that keeps all 100 enzymes happy. But what you could do is you can split it. If you wanted to do 100 enzymes, maybe you need 5 buffers. So you do 5 reactions. Then you can combine it and then shoot. You wouldn't have to shoot 5 separate into the mass spec. You can combine them all and shoot the whole thing and analyze all 100 at the same time. So I think that's possible. The problem is we don't have treatments for 100 diseases right now. So the beauty of this method is as treatments develop, we can just add another domino to the stack, right? We can just keep adding more and more assays. And that's the beauty of the method, right? And it wouldn't be much harder to add on another one.
Do you have plans to introduce technology for measuring enzymes that are not in lysosomes, that would be in other parts of the cell?
It would work, I think, it would work on any enzyme that's happy to be dried down into a blood spot, I think. I don't see a problem. Yeah, we're working on other porphyrias, for example. Porphyrias, biosynthesis of heme is a problem. These have been a nightmare to diagnose, apparently, but there's no treatment for it, so it's not for newborn screening, it's for diagnosis. But yeah, we're working on a lot of different enzymes other than lysosomal enzymes, but for newborn screening, has to be treatable diseases, right? For the time being.
It's certainly helpful.
Yeah, sometimes it's useful to know that you're a carrier, right? And that your wife's a carrier, but it's just not, it's very controversial to screen for everything, right, obviously. So we start with the most obvious.
Great, thanks.
Thank you all for coming. So.